Hello again. In the previous video, I mentioned about powerful genome databases and browsers that store and allow analysis of the genome sequences, and it also provides um, graphical user interface that's easier, that makes it easy for the average scientist or even the layman to understand. This table 7.1 gives you an example of the different databases, and this is just a glimpse, right, of what information is available. It says here, for example, this is the human chromosome 7, if I'm not mistaken. It's got the CFTR, the cystic fibrosis transmembrane regulator gene, and it spans 117465784. What number is that? This is the number. When they start sequencing for each chromosome, they'll number at one, and any particular segment at 1 to millions and so on. So this is the 117,465,784th base until 117,715,971 based on chromosome 7. That is the position of the human CFTR gene. And look, what they also have now is that each gene will have the sense transcript, which is the mRNA, and then it will have also the antisense transcript, which presumably does the regulatory function, and also it shows the transcripts from neighboring or um, adjacent genes, partially overlapping protein coding genes, the CTT and BP2 transcribed from the opposite DNA strand. So it is referring to this right end here. It says the transcript actually overlaps with the CFTR coding sequence on the other strand. Um, um, Exons are represented by the exons are this vertical bars. These are the exons, right? Very, very small exons, and a lot of them are actually non-coding. The exons are the coding, and then the lines, this upside down V, the chevrons, upside down V is called chevrons. These are the introns. So that's the exon, intron, exon, intron, exon, intron. The 5 CFTR protein coding transcripts, 2 full-length isoform and 3 smaller isoforms, 6 non-coding sense transcripts, and 2 antisense. So this is why one of the part of the reasons why the exact number of genes is at the moment quite impossible to be determined. Okay. Bioinformatic approaches to predicting genes and their functions. So it is now established that getting the sequence is the easy part. Next is to identify the genes and also to annotate its function, and that has relied a lot on bioinformatics. So one of the first things to do is that we know they know of the existence of some genes, but a lot of the genes are not yet found. So at that time, the strategy that they used to identify genes were based on several approaches, and the two biggest approaches are prediction based on certain characteristics. Now, for example, regions that contain genes and regions that contains non-genes have different sequence behavior. The non-gene sequence have high A's and T's. Even the stop codons, right? The UAA, UAG, UGA, it contains more A's and T's compared to the coding sequences. Coding sequences also, there are certain codon biases that can be taken into account. There are a coding region also has translatable sequence um, of a certain length, normally more than, and any sequence that can be translated, maybe for 15 or 20 amino acids is can be considered as a coding sequence. So all these characteristics can be taken into account and be included in the design of algorithms to predict whether a particular sequence contains genes or not. The other way to identify genes is by looking at similarities. Remember, the Human Genome Project is not just about getting the sequences from the human genome, they're also getting sequences from accompanying genomes, the E. coli, the yeast, and so on. So what they do is they start to compare, and because obviously the microbes, the yeast, they are smaller genomes, and they are much easier. They're not as complex as the human genome. So understanding them is much easier, and because of that, they can be used as a reference, as a resource, as clues, in order to understand the human genome. And rightfully enough, a lot of the things that we study, the basic processes in the human genome, has always been first studied on the microbes. All right. So hidden Markov models. In order to understand that, you'll have to do some readings on statistics. Basically, it's just saying that some some occurrences may appear random, but the randomness is actually controlled by some factors. Hidden Markov models. I will not explain here because I'm not that good to explain about statistics as well. So if you're interested, you can read up. Annotation. Annotation is the key word nowadays when we talk about understanding the human genome. The next is to annotate. Annotate means you don't want to mark where the genes are, where the coding sequence starts, where are the intron exon boundaries, and where is the promoter sequence. This is annotating the genome. So this is what they do. And gene ontology. Okay, it says that the sentence, if you read it, it says, as function became mapped to genes, collaborative efforts sought to annotate genes in a comprehensive, systematic way, and the genome and ontology project was developed. So the GO. So at the moment, if you do bioinformatics, you can't run away from doing GO, gene ontology. That means looking at the function of genes in a network. Okay. I think before I go on, I'll stop here, and then I will see you again in the next video.